are streaming. Okay, good.
This is me changing the streams.
Okay,
But this is good that it's working.
Yeah, it's good.
It's on the big screen. Nice. Okay, I'm gonna open my computer. Make sure it's actually working.
Oh, to see the
Sound.
Okay.
That's it. But it's probably gonna take like. Oh yeah.
Oh, okay.
This is me changing the streams.
Okay,
But this is good that it's working. It's so funny. Okay, I'm gonna open my computer. Make sure it's actually
Oh, to see
sound. That's it. But it's probably gonna take like.
The suppression happens right after this. Like so first facilitation.
Oh, okay.
It's literally like
This is me changing the streams. But this is good that it's working. It's so funny. That's it. But it's probably gonna take like.
the suppression happens right after this. So first facilitation.
Oh, okay.
It's literally
desktop one?
Desktop.
Oh,
Maybe it's interfering.
yes. Maybe it's interfering. Well, at least this microphone is working.
Oh, it sounds good. I think it worked. Am I interfering? It worked. I'm so bad with technology.
Your fury.
I'm gonna teach myself Python for summer. It's gonna be a good time.
But I actually don't know. Okay. This microphone is working. Okay, hi everyone. Sorry to startle you. I think we're going to get going if you don't mind. We do have a lot to cover today, and there are some techniques that you need to be familiar with for the final exam, and so I'd like to make sure that what we're covering that you also are going to be responsible for on the quiz is material that you understand and can demonstrate that you have understood it. So one of the things I also want to talk to you very briefly about, some of you who are in fourth year or are thinking about graduating are thinking a little bit about grad school and about research. And one of the things that I can recommend to you is to really be careful in picking your supervisor wisely before you decide to go into research, because it is a long-term commitment, whether it's a master's degree in 2 years or slightly over 2 years, or a PhD where it's going to be 5 years. And one of the things that I didn't like about my master's experience was I wasn't used to the environment. I didn't really enjoy my research, nor did I enjoy the lab that I was in at the time. And for the 2 years that I was there working on my master's degree, can you imagine going into work every day and actually not enjoying it? And you're there for about 12 hours a day at some points in time. So one of the things that I'm dedicating this lecture to, for a variety of reasons, one of which you'll understand a little bit later. When I started my PhD, I was really careful. I wanted to work with someone who was much younger than my previous supervisor, someone who was going to allow me to talk about things that I wasn't sure about, to give different ideas and opinions on things. And one of the supervisors that I worked with, Dr. Yutian Wang, who went out to UBC, actually was all of those things. He was very supportive. He was really young, so he understood what it was like to come out of grad school and to understand what my needs were as a graduate student. So again, this lecture is dedicated to him, not just because he was a fantastic supervisor, and I want you to find a fantastic supervisor, but also for reasons that I'll get to in a few moments. So we're going to be going back and talking a little bit about LTP and the synapse and the modifications that occur at the synapse. And I'm going to go over some of the material that I talked about in the last lecture as well. So just a few reminders for those of you who weren't able to do online quiz 1 because you were ill, etc. It'll simply be reweighted to the final exam. So that 2% is something that you should be mindful of. For online quiz 2, it'll start at exactly the same time. It will run until Saturday morning. It will be based on this lecture as well as what I cover on Wednesday. It'll all be based on what is covered in lecture, so please make sure that you have your notes ready, that you're giving yourself about 20 minutes to be able to complete the 10 questions that are online for you later this week. If you are writing the deferred term test, that will be written on Friday at 3 o'clock. You should be getting a notification from the HMB office of the location. If you don't get that by Thursday, please contact the HMB office directly. I can also tell you that I have received about half of the marks for the short answer questions for term test 2. And those of you who are coming in to view term test 1 today, maybe I'll chat a little bit with you about term test 2 at the same time, depending on when the marks are sent to me by the TAs. Okay, so I'm going to go back and we're going to revisit different aspects of LTP. I know you've had a number of lectures in some courses on LTP, and I wanted us to cover it in a slightly different way. I wanted you to understand why we think we know things about LTP and how we understand those things related to long-term potentiation and why we think receptor trafficking underlies that whole phenomenon. Nowadays, because I looked in a textbook just the other day, we tend to take for granted that LTP and LTD involve the trafficking of receptors, and we know that really well in 2019, but I wanted you to get the understanding and perspective of how we developed that story over the last 20 years. So we'll be going over a number of these different types of experiments that relate to subunit trafficking and receptor trafficking, why we think LTP underlies learning as well as memory and the strengthening of synapses, and we're going to use a particular model system to be able to do this. So in the last lecture, we had talked about how LTP, long-term potentiation, long-lasting changes, not the short-term changes that we can observe immediately after stimulation, so that would be things like facilitation, augmentation, even post-tetanic potentiation is considered relatively short-term, but if I stimulated a synapse or stimulated 2 neurons, and then I went back 1 hour later, would I still see a strengthening or a weakening of that synapse that we think was being activated? If we do see that, that is what we call long-term LTP. Most of you have learned it in the sense of homosynaptic LTP, one synapse, and how that one synapse gets strengthened. I also indicated that the other type of LTP is known as associative LTP. So not just that one synapse is getting strengthened, that homosynaptic form of LTP, which is down here up here at the top, but associative LTP, where on a different input, we can also see strengthening just because it happens to be firing at the same time. So if we get 2 different inputs, one where it's repeated, but at the same time, if I have a different input into the same group of neurons, I might also get associative or paired LTP on that synapse as well. We now know that whether we're looking at the first type that all of you are familiar with, repetitive activity and the strengthening of this particular synapse, homosynaptic LTP, or this synapse, which is also being strengthened, which we're calling associative LTP, it just happens to be going along for the ride. Both of these forms of LTP require the receptor NMDA, so the N-methyl D-aspartate receptor, as well as the AMPA receptor that I'm going to be talking to you about in a moment. And one of the things that we know is required for this is that there has to be a baseline level of overexcitation or activity to allow for NMDA receptors to become activated. And why that's important is that the NMDA receptor at rest is blocked by magnesium, and this dual activity, this paired input, allows for the magnesium ion to be released. We need that depolarization through this sustained activity at the associative synapse in order for the magnesium that is blocking the NMDA receptor to be removed. And again, I'll explain that to you in the next slide, but this idea and this concept that we need NMDA receptor activity and that we need depolarization first to remove that NMDA receptor blockade is a really important one. We're going to see this several times today. You're going to see it on your quiz, and you're also going to see it on your final exam. And this idea that this receptor is maybe the molecular basis for learning as well as memory is a really important one. And the reason why we think that this NMDA receptor is so important, especially for long-term changes, is that the one ion that we need to enter into the cell is calcium. And calcium can only flow through in the normal physiological brain through the NMDA receptor. So we need the NMDA receptor open. It's usually blocked. We can unblock the magnesium that's stuck inside the NMDA receptor with sustained activity or depolarization, and that step is critical if you're going to learn, remember, or be able to associate different ideas in terms of LTP. So all of these things together suggest that any form of LTP that we're looking at, whether it's homosynaptic or whether it's associative forms of LTP, when we go back and look at the molecular correlate, what allows for this to occur? So for associative LTP, for example, we need the cooperation of two different synaptic inputs. We need input 1 and input 2. So these have to be coming in together. We also need to know that only the synapses where we are getting action potentials are going to be strengthened. So if I had a third synapse and it wasn't firing at all, so input 1 and input 2 are firing together, but input 3, a third synapse, if it's not active, it will not get strengthened. There has to be some kind of input specificity, and there has to be some sort of molecular mechanism that allows for this activity to be associated with this long-term change. So something about the activity has to work on some kind of receptor system, and I've already told you that that receptor system is the NMDA receptor, or the N-methyl D-aspartate receptor that is required for LTP. And in the 1980s, this one receptor was found to meet all of the different things that we needed for LTP. It explained cooperativity because the magnesium block. It tells us about input specificity because of how these synapses look, which I'll get to in a moment, as well as having sustained activity and being able to unblock that magnesium from the NMDA receptor. So again, you need to know this slide. You'll be asked quiz questions, as well as for the final exam, you'll need to know details of this slide and the next few that are coming up. And you'll also need to understand what's on this slide for what I'm going to talk to you about later on today in lecture as well. So if we look, what we're talking about right now in terms of long-term changes is really about the glutamate receptor. I told you earlier that can we see the same thing for inhibitory GABAergic receptors? And we can. But for the most part, 80% of your brain involves these three types of receptors. So it involves glutamate. So whether glutamate is working on the NMDA receptor, or whether it's working on the AMPA receptor or on the kainate receptor, glutamate will bind to these receptors, and 80% of your brain uses some form of these receptors for glutamate neurotransmission. So these are also found on the postsynaptic side of the cell, and these are, again, very important because these are all cation channels. The ones that I'm going to be concentrating on for the most part are the AMPA receptors as well as the NMDA receptors, because usually at most glutamate-containing synapses, so glutamate receptor-containing synapses, the vast majority of them will contain both AMPA as well as NMDA receptors. Now just keep that as a caveat for the time being. There's a little bit of an asterisk or an idea that you have to also think about in the upcoming slides as well. So the NMDA receptor, as is outlined here, is specialized because if you were to take a look, even though different types of cations can flow through the AMPA, kainate, and NMDA, NMDA is the only one of the glutamate receptors that after binding glutamate will allow the entry of calcium. None of the other glutamate receptors will allow this to occur. Again, with a little asterisk and a little caveat associated with this. Okay, so this is an important slide for you to make sure that you know. As you can sort of guess from the topology, we usually have 4 subunits. So this is subunit 1, subunit 2, 3, and 4. All of these glutamate receptors that are ionotropic, that allow ions to flow through, all of them have 4 subunits that are associated with making up the receptors. So AMPA, NMDA, kainate are all heterotetramers. Again, a fact that you need to know and is also outlined on the next slide. So again, the vast majority of the receptors we're looking at are the NMDA receptors, and then the AMPA receptors. I'm just going to call them AMPA receptors because even though kainate is found in certain regions of the brain, for the most part, we think LTP is due to AMPA receptor movement that I'll get to in a moment. So again, this is a really busy slide, but I'm hoping by the end of today's lecture, especially in hour 2, you'll understand why we went through this. So why are we talking about this NMDA and glutamate? When we are causing the presynaptic release of glutamate onto this postsynaptic cell, and we are measuring LTP after 1 hour, is this synapse strengthened? After glutamate has been released repetitively over a short period of time, can we see after 1 hour any strengthening of this postsynaptic cell? So when we're looking at this cell, we are looking at how glutamate gets released. Glutamate releases onto both NMDA as well as onto AMPA receptors, kind of shown here as a non-NMDA receptor, but we'll call it the AMPA receptor. So at rest, when there is no change in the membrane potential, which is why you're seeing low voltage here on the right, low cell voltage at rest, it is blocked by magnesium. That is a fundamental property of the NMDA receptor under resting conditions. A couple of other things to make note of as well. So when we have these glutamate receptors, even though they're all heterotetramers, and even though they all kind of look similar to each other, in fact, we can actually use different types of toxins and drugs to differentiate between the NMDA as well as the AMPA type receptors. So the NMDA receptor, for example, can be blocked by drugs such as APV or AP5. It can also be blocked by MK801, which is an open NMDA receptor channel blocker. So it has to be open first, and then MK801 can block it once it's open. And AP5 and APV are normal antagonists for the NMDA receptor. It prevents the binding of glutamate to these types of receptors. Another type of blocker is CPP, which is another antagonist for the NMDA receptor, and it prevents the binding of glutamate. If glutamate doesn't bind, it cannot open this receptor. The last thing that I'll make note of as well, we have a very special drug, which is known as ifenpridil. And ifenpridil is an NMDA receptor blocker, except there's one condition associated with it. The NMDA receptor has to have a specific subunit. It has to contain the subunit which is known as NR2B or GLUN2B under the new nomenclature. The old name used to be NR2B, and you will still see papers that use NR2B as a subunit. This means that it's NMDA receptor subunit 2B. The new nomenclature is the glutamate NMDA 2B subunit. This can be blocked specifically with ifenpridil. We have other drugs that will only block the AMPA or the kainate type of receptors. We don't have many drugs that can actually separate out AMPA versus kainate, but in general, when we're looking at AMPA receptor blockers, we will use drugs like Geikie, DNQX, CNQX, NBQX, etc. So anything with QX, usually is an indicator that it is an AMPA receptor blocker. So we have these different drugs that allow us to differentiate between NMDA receptors found on the synapse as well as the AMPA receptors that are found on the same synapse. So glutamate will bind to both of them. And how do we know that it's AMPA that's changing and not NMDA? These drugs actually help us to determine if it's a change in AMPA or if it's a change in NMDA. So again, just before we get into all of these details, we are looking at long-term changes. So we have repetitive activity, and when we're looking at long-term changes, we're always looking at repetitive activity no matter what you look at. So even if you're thinking about associative LTP, the other synapse still needs repetitive long-term activity, and you need to pair that with at least one action potential. So regardless, LTP always requires repetitive activity, and most forms of LTP, especially within the hippocampus, requires that we have NMDA receptors, and these NMDA receptors will allow for calcium to flow into the postsynaptic neuron. As I mentioned to you before, at rest, these NMDA receptors are normally blocked by extracellular magnesium, and this magnesium can be removed if we change the membrane voltage. If we depolarize the neuron, we can then remove the NMDA receptor block that is caused by this magnesium. And these NMDA receptors are thought to be coincidence detectors because they require depolarization as well as the release of glutamate binding to the receptor. So when you have presynaptic activity and you are working on having glutamate released, because it's nearby to the AMPA receptor, this is one of the ways in which the AMPA and the NMDA are both detecting at the same time the release of glutamate. And I'll go through the mechanism in some more detail in the next few slides. The same signal, in other words, are coincidentally activating both types of receptors. So again, shown to you here, this is what we're looking at. We are looking at this particular synapse here on the left, and this axon is repeatedly releasing glutamate. We're doing that high train of activity. All of those action potentials that I showed you last week, we're causing the release of the neurotransmitter glutamate, denoted here by G, and it is acting and binding to both the AMPA as well as the NMDA receptor, and at rest, this NMDA receptor is normally blocked by this magnesium. However, at some point, when the glutamate binds to the AMPA receptor, it is going to open the AMPA receptor and allow sodium to enter. If it's entering this cell here, what will happen to the membrane potential? So positive charge going in, the intracellular charge will change. It will become depolarized compared to what it was before. And this depolarization will then allow the NMDA receptor to become activated in the presence of glutamate, and that magnesium will get released. And now we have the first entry of calcium. And this is what will account for the long-term changes that we see in the synapse after repeated activity. So before we get into how we know that it is AMPA versus NMDA, there are a few things that I needed to take a little bit of a sidestep and a sidetrack to get us on the same page. Early on in development, when we are starting to form these different types of synapses, there is the concept of having what is known as a silent synapse. And this silent synapse looks almost identical to a mature synapse. So this would be early in utero development, so before you are born. You would have a number of these different types of synapses that you see here. So we still have glutamate presynaptically. We can still stimulate this presynaptic, the presynaptic axon. So if we fire an action potential, the glutamate will still be released. But if we were measuring from this cell down here at the bottom, we would actually not be able to see any type of activation. We would actually see that there is no change. In fact, we call this synaptic failure. Why that occurs is that we only have the NMDA receptor. Early in development, we don't have both AMPA as well as glutamate. And that's why it's known as a silent synapse. The synapse is there structurally, but because there's no AMPA, and because when glutamate gets released, there's no way to depolarize this membrane, this is known as a silent synapse because even in the presence of glutamate, we would never see this neuron here on the left firing. We would have to do something different to allow for this cell membrane to become depolarized and allow for the entry of sodium as well as calcium. Again, it's not shown to you on this particular slide, but can you think of what receptor might allow you to do this early in development? There is no AMPA yet. AMPA does not exist. It gets turned on transcriptionally later on. Any ideas? Yes. Acetylcholine. A good idea, actually, because acetylcholine, if it were binding, would actually allow for the release of or allow for depolarization. It's actually a really good idea. It doesn't occur here. This is in the brain. It's all over the place. What do you think it might be? And it's a little counterintuitive. How could I depolarize this cell membrane at rest very early in development? So one of the things that we have, and it's a little counterintuitive, is we have a lot of GABA A receptors. And these GABA A receptors are normally inhibitory. However, when your neurons are really, really young, before they form these full-fledged types of receptors that you have here in panel B, when we're looking at what happens early in development, in fact, when you get the release of GABA early in development and it acts on GABA A receptors, it actually causes depolarization, not the hyperpolarization that we see in mature neurons. And so we know that these neurons can become functional and they do respond and you can depolarize them, but you depolarize them actually with GABA instead of glutamate. They're not true coincidence detectors because GABA is a different neurotransmitter than glutamate, but we can do that sort of artificially in early development. Later, though, we will have both AMPA, shown here on the right, and NMDA, and so we do have coincidence detectors, but early in development, we have silent synapses. This is a really important concept because think a little bit about this. What this is suggesting is somehow AMPA receptors can be inserted into the membrane of the postsynaptic cell, and this is the idea that scientists were working on in the late 1990s. Are there silent synapses, and can this explain LTP? And I'll get to that in a few moments. So one other thing to make note of, and again, you need to think a little bit about these slides. I'm going to tell you that you will have questions related to this on your final exam. I know how much you love IV curves because that's all you think about all the time. And these IV curves are actually now things that you can think a little bit about and be able to critique them in a little bit more detail. So here is what a normal glutamate response looks like. This does not look like any of the IV curves that you looked at earlier. I'm just recording from a postsynaptic cell at different membrane potentials. So at minus 100 millivolts, at minus 60 millivolts, minus 20, and then plus 20 millivolts. This is in the presence of glutamate, this is how the postsynaptic cell responds. So this is the glutamate response. And what I'll see at these different voltages is this peak early response and then this late phase response here. And at these negative voltages, so minus 100, minus 90, minus 80, etc., the early response actually dominates most of the recording. So you can see that this is the biggest part of the recording. The late phase is almost non-existent. And as we continually change the voltage and we look at the resulting currents that result, so these are all currents that you are seeing as these traces, one of the things that we would begin to see is that this changes and the late phase will change as well. And we can map out this IV curve, and this is what the IV curve actually looks like. The IV curve has two different components. There's an early component and a late component. And if you're taking a look, and again, making sure that you're all aware and you have reviewed this, what is the reversal potential for both AMPA as well as NMDA? So based on the IV curve that is plotted here on the right, what is the reversal potential? So you're all thinking 0, and you'd be right. For both of these different receptors, whether you're looking at the early current or the late current, the potential, the reversal potential is 0. In fact, the IV curve goes through 0, as you're seeing here. But what this also suggests is that if we're looking at the early in red, this is more of a straight linear line. And that is because this is the AMPA receptor. This early peak current is due to AMPA receptor activity. And if we look at the AMPA receptor, it has this linear sort of IV curve, and that's because it is really conductive to single charged positive ions. And you have to, again, think a little bit about this. Based on where it's going through, should you expect to see the reversal potential, thinking that it has physiological extracellular sodium, extracellular potassium, calcium, etc. Everything is done under physiological conditions. Should it actually go through 0? It actually shouldn't. If it was only sodium, as is shown to you on most of these diagrams, sodium enters. Again, on this diagram, sodium enters as well. If it's only sodium, you know what the reversal potential should be. It should be around plus 50. And what this is suggesting is that more than one ion flows through. That other ion happens to be potassium. Normally, we don't really think too much. But if you're thinking about why it's going through 0, it's because more than one cation actually flows through the AMPA receptor in red. And the NMDA receptor, we have sodium, calcium, and potassium flowing through that when it's open as well. So again, you should probably make sure that you are familiar with the IV curves for AMPA, which is linear. And for the NMDA receptor, we still have an inward current at these lower voltages, and then it reverses, and then it will actually start to flow outward. So one other thing to just get through some of the technical details. I mentioned to you earlier that we have a number of different blockers, and we can do these types of recordings that I've just shown you. So I just showed you on that previous graph the EPSCs that result when we voltage clamp at minus 80. This is what results. The voltage clamp at minus 40. And then at plus 20, this is normally what we would expect to see up here at the top. I also told you that we have a number of different blockers for different types of glutamate receptors. Without referring back to your notes, because this is short-term memory, what does APV block? So APV is normally a blocker for the NMDA receptor. So if we are doing these experiments where we're doing this IV curve, and we have the presence of APV in the recording solution, and it's blocking the NMDA receptor, one of the things that we see is APV has no effect at all on the early current, which is, again, what you'd expect. The early current is this peak value here. We don't see any change, but we see that the late current, so up, this is what it should be normally at the top, and it drops all the way down here at the bottom. So if you're thinking about this, one other factor that I didn't mention to you earlier, where do we see the late current the best? So at negative voltages or at positive voltages? The NMDA receptor mediated change, we can see it best sort of at the plus 20 millivolts, etc. And we can see the effective APV much better at these depolarizing voltages. We see it a little bit at minus 40 as well, so that late current is actually also affected. So we can do the same thing on the same group of neurons by also applying CNQX. So CNQX, going back, would I see the same effect if I applied the results that we see here on the right? If I use NBQX, would I see the same result, do you think? How about if I use something like DNQX? If you're saying yes, then you would be correct, because these drugs here are blocking the AMPA receptor, and you can see that this early current goes down to nothing. It disappears entirely in the presence of CNQX. This early current is also affected here, and there's a small change because usually it doesn't dominate at these positive voltages, but CNQX also has a small change on the AMPA receptor mediated current at plus 20 as well. So those different types of antagonists can actually tell us which parts of these currents are actually changing when glutamate is being, when we're looking at a glutamatergic synapse. Okay, so we've just talked about an awful lot of different things. Do you have any questions on any of this before we go on? Whether it's the IV curves, the different types of antagonists, whether it's how this all works. Any questions? Yes. Yes. So I think your question is, until it gets depolarized, will there be any sodium entry into the cell? There will not be. So under normal circumstances, it's blocked by magnesium. So we can get around that by doing these types of voltage clamps, or we can change the amount of magnesium so that magnesium isn't blocking it under resting conditions. Okay? So it's a really good question. Yes? Why is it more pronounced for this early current? So again, a really important concept. You'll be asked questions. Again, I want to be completely transparent about it. You have to understand what these are actually showing. So it's a little like this. Glutamate is going to work on your postsynaptic cell, but glutamate binds to two different things. I can't tell the difference. So glutamate will bind to AMPA. It'll also bind to NMDA, and it gives me this global response. And this global response looks a lot like this. But there are actually two different phases to it. So there's an early acting response that's due to glutamate binding AMPA receptors, and that's the early phase. And then there's a slower component that is due to the NMDA receptor binding to glutamate and being open. So why do we see CNQX working here? Because it is blocking the AMPA, which is always acting early. Does that make sense to you? I'm not sure if it does, but I'll assume that it does. Are there any other questions? It's a really important sort of discussion. You need to make sure that you know it. If you have any questions, please make sure that you ask, because I can tell you, you will get these types of questions on your final exam. No? Okay. So, as many of you have kind of picked up in other courses, and if you haven't, that's okay too, because we're going to discuss this right now. Many of you will have learned that after LTP, after this high train of activity, if we wait 1 hour and then we go back and examine this synapse, if this is what the synapse looked like before, and it has both NMDA as well as AMPA receptors, one of the things that we can see after LTP, after this high-frequency stimulation, after trains of action potentials have been delivered through this synapse and lots of glutamate has been released from these different vesicles, then one of the things that we'll see is postsynaptically, this particular synapse that we're looking at here, and again, just looking at this and based only on this diagram, would you think that this is homosynaptic LTP or associative LTP? So you only see one synapse, so this would be homosynaptic LTP. One of the things that we would see is there's an increase in AMPA receptors after stimulation has occurred. And you've seen that over and over again, and you have this idea that AMPA receptors, there's more of it on this postsynaptic membrane, and we get more intracellular calcium, etc. And this is, again, a really important consideration as well. And this is sort of the model that all of you have learned. How does that all occur? So after stimulation, after glutamate gets released, after NMDA receptors and AMPA receptors get activated, how does this all work? So one of the things that we think happens is that these NMDA receptors that work as coincidence detectors, both AMPA and NMDA, coincidentally detect glutamate release. AMPA receptors cause membrane depolarization because they allow sodium to enter into the cell, and this will then release magnesium from these NMDA receptors, and then we get this entry of calcium when glutamate binds on these NMDA receptors. And this calcium entry through the NMDA receptors will then cause it to bind to calmodulin, and calmodulin will then activate through the calcium calmodulin dependent system, and will activate CAM kinase 2 in this particular spine on this synapse, and this will then activate this CAM kinase 2 through autophosphorylation. It phosphorylates itself. And then beyond that, we also think now that this activated CAM kinase 2, which has only been activated after NMDA has been turned on, will cause more intracellular AMPA receptors that exist in these vesicles to traffic up to the plasma membrane, putting more of them on the plasma membrane. Or alternatively, or in addition to, where again the literature is sort of split on this, CAM kinase 2 may also cause the phosphorylation of AMPA receptors that are already on the cell surface, in addition to causing more of them to traffic. And this phosphorylation of AMPA receptors means that in response to glutamate binding to the AMPA receptors, after phosphorylation has occurred, the AMPA receptor stays open longer. The mean open time, something that we talked about many weeks ago, the mean open time increases for these AMPA receptors in the presence of activated CAM kinase 2. All of these things say, therefore, that in response to the same amount of glutamate that it saw initially, before stimulation had occurred, before LTP, the exact same amount of glutamate will now cause a much bigger response here in this postsynaptic cell. And again, this is something that I glossed over in the previous slide. If you take a look, based on everything we know in 2019, there are two different mechanisms for how this synapse can respond. One, it could just be releasing more glutamate than it did before, or it's responding to the same amount of glutamate in a much more robust manner. And it turns out that it's not a change in the amount of neurotransmitter, it's really the response that is important. So this is where we're sort of at. And this is, again, a lot of molecular detail that you probably have learned about in several courses. And I wanted to also, before going on, talk about the other process, which is known as long-term depression, or LTD. If LTP involves the insertion of AMPA receptors, LTD might be the removal of AMPA receptors from the plasma membrane. Again, for a variety of reasons that we're not going to go through in great detail, I'll go through one more slide related to this. LTP and LTD both require NMDA. You have to activate NMDA. Again, it seems counterintuitive, but when we have activity, we will get the release of glutamate, and then it will act on the NMDA receptors. These NMDA receptors will still allow calcium to flow in to this postsynaptic cell, and this will then activate a number of other enzymes such as phosphatases like PP1 or protein phosphatase 1, which is the long version of PP1, or protein phosphatase 2A, as well as calcineurin. Phosphatases get activated in LTD versus kinases, which get activated in LTP. And this causes the removal of AMPA receptors from the synapse. But this, again, is a really bizarre idea, and it's really conceptually difficult, and scientists struggled with this for well over 10 years to try to come up with an explanation. Because think a little bit about this. So imagine, for example, in one course, you have studied, you've taken great notes, and you've done everything that you can, and then you went out, you wrote your test, and it turns out that you got an A+. And let's say you do exactly the same thing, but now when you write the same test, you end up getting something like an F. How would you be able to come up with an explanation that links these two? And here's why they are very, very similar. So this, again, is LTP. So LTP is shown at the top. So this is the normal baseline response. We stimulate with high trains of activity, which we call tetanic stimulation or high-frequency stimulation, where we deliver 100 action potentials per second. We then wait a little bit. And then we go back and measure, and we see this really strong increase. It has increased by 50%. It's gone from 100 to 150% of the response that it had before. It is also very specific because if we took a different synapse and we looked at its effects, we would actually see that there's no strengthening. It is very input specific. But look down here at the bottom. Low frequency stimulation on the exact same synapse, and low frequency meaning that you have 1 hertz action potentials or 1 action potential per second. It's still fairly rigorous and it's still fairly a lot of activity. This actually causes a weakening of the synapse. So how do you account for overactivity and still elevated activity more than you would have normally at baseline, causing either a strengthening on one hand or a weakening on the other? It's again going back to that same scenario. You've done everything almost identically. It's still a lot of activity, even 1 hertz, and yet we get totally different and opposite results. And this bothered scientists for a really long period of time. We're looking at the same group of neurons. We're looking at the same sorts of synaptic changes. We know it involves NMDA, but how do we account for this change? And again, this is a really busy slide, but I am going to ask you to know just 2 details from this slide. So this illustrates exactly the same thing. So low frequency stimulation versus high frequency stimulation. So on the left, we will have LTD. So this is outlining LTD with low frequency stimulation versus high frequency stimulation here on the right. We are looking at the same synapse, if you want to think of it that way. It still requires the activation of the NMDA receptor in purple, and it still involves changes to the AMPA receptors overall. I've outlined for you the pathway here, that it requires calcium entry. It still requires calmodulin dependent processes. Here, these are all phosphatases. But how do we link these 2 things? What's the critical switch that allows us to differentiate between having either LTP or LTD in the same synapse? So how does it know, if calcium is really the key indicator, how does it know the level of calcium? How does it know which pathway it should take? So the key thing in this particular pathway, and one of the things that seems to be important, is that if we look at sort of the alternative pathway after calmodulin activation, it can also stimulate adenylate cyclase in this same cell. And this is after high frequency. We do not see activation of adenylate cyclase with low frequency activation. And this will increase intracellular calcium, cyclic AMP. And this increase in intracellular cyclic AMP is also very specific to high frequency LTP only. We never see this in low frequency. And this in turn will activate protein kinase A or PKA only after high frequency. So this is sort of the rate limiting step. This pathway here allows for the neuron to know if it should be undergoing weakening or strengthening when there's a lot of calcium intracellularly. And some of this is related to where these different systems are located, et cetera. And the other key factor in LTP versus LTD is this other switch as well. Both of them converge on this particular process. And this other inhibitory 1 protein, or I1 protein, and whether or not it gets phosphorylated or not, really determines if we get LTP or LTD. Again, I'm not going to go through all of the mechanisms here, but the fact that we will see this pathway only activated in LTP, never turned on in LTD, is important. And the key role of this inhibitory or I1 protein allows for the neuron to know if it should be undergoing LTP or LTD. And again, we won't go through all of the details of all of the experiments that have been done to verify that these are the critical elements that the neuron has to know. Ultimately, though, we know that there are these different pathways, ones that you should make sure that you know for the LTP versus LTD pathway. Again, this is still very, very controversial. even after 20 years, even though this is the best molecular mechanism in all of neuroscience, and we know all of the different details down to the last amino acids, there's still a lot of controversy that exists about whether or not all of these different things are required all the time. And I think I'm just going to go through this one slide and then we'll take a break. And again, this is another slide that you should expect to see on your final exam, as well as on your upcoming quiz. Just a little bit of a heads up for those of you who are doing the quizzes. Please make sure you're starting early. I don't know what's up with your class this year, but about half of your class actually started the day of, half an hour before the actual deadline. And I don't know if you like living dangerously or not. If you get that really dopamine release and you really like that dopamine release, please don't do that. It really gives me a heart attack kind of watching this spike. Everyone's logging in on Saturday morning. It's like 10:30, 11 o'clock, and I hope they all make it. And some of you did, some of you didn't. Don't do that again. If you start early, it's always better for you. I can also tell you that some of the questions from the quizzes that you get this week will be very similar to what you get on the final exam. I will be posting those for you as a PDF document, so you don't have to do those screen captures that many of you cough might be doing. So I will provide all of those for you as well. So just a couple of things. All of the receptors are heterotetramers. NMDA receptors are heterotetramers, AMPA receptors are heterotetramers. And then if we're looking at AMPA, the old name is the GLU-R. Nowadays it's GLU-A, but they have 4 different subunits: GLU-R1, GLU-R2, GLU-R3, GLU-R4. Every one of the subunits has the same topology that I'll show you on the next slide after break. So GLU-R1 has the same topology as GLU-R2, which has the same topology as GLU-R3 and GLU-R4. These different subunits all have intracellular C-terminal domains or carboxy tails, and all of these different carboxy tails have sites for phosphorylation. So they can be phosphorylated by CAM kinase 2, can be phosphorylated by protein kinase A, etc. And again, another thing to make note of, and this is why this question is being asked. So if NMDA looks almost identical to AMPA, what is the difference between NMDA and these AMPA subunits? So they look almost identical. Why does one allow calcium entry and the other one does not? And it turns out there's one reason, because 99% of all of your AMPA receptors that exist within your brain, every last one of them, or almost every last one of them, contains GLU-R2. And this is the reason why we need NMDA receptors. GLU-R2 that is found in almost every AMPA receptor that you have in your brain is actually unique and ubiquitous through RQ editing, which is at the RNA level. That means that every single AMPA receptor early in development had an RQ switch, a glutamine and arginine switch, and this makes it impermeable to calcium. Early in development it was, then it switched over and it became impermeable to calcium. So AMPA receptors actually, because of the GLU-R2 subunit and because the GLU-R2 undergoes RNA editing, is completely impermeable to calcium. And that's why we need these NMDA receptors. So I think we've just covered a lot of material. It was pretty heavy material, so I think we'll probably just go ahead and take our break now. If you do have any questions, please let me know. I'll be happy to try to answer them or answer them after the break.
Question regarding this slide. So we need AMPA to become activated to activate NMDA. So if you block AMPA, how do you still get NMDA activated?
So here we are providing the change across the membrane.
Oh.
Remember, this is voltage current.
Okay, okay.
So it's an artificial system.
Okay, thank you.
If we want to determine that the NMDA receptor contains NR2B
Oh,
subunits,
just because. Okay. Okay.
still some activity, so it's as if that there's was activated through the organization. Does that cost something? I'll just about maybe about a talk about child children dying and women being separated from their babies. I asked for extension of the kids. Yeah. I'm pretty much, yeah, it's very similar. My topic was like, was continuous life. And I had all of my reviews, I like that.
Okay, hi everyone. So a lot of really good questions during the break, and I will try to speak a little bit more slowly for those of you who want me to speak a little bit more slowly. Just a couple of things about this slide. You do not need to know all of the details that are found on this particular slide. So everything down here at the bottom that I didn't go through, if you're worried about what a PDZ binding domain, what SAP97 interaction proteins look like, you do not need to worry about those. I'm going to go through what is here on the right in blue with you as well. The NMDA receptors are almost identical to the AMPA receptors that I went through on the left. They are all heterotetramers as well. And these are very similar, but they're called GLU-Ns rather than NR1, etc. Just like GLU-R2, or down here at the bottom, is ubiquitous. It's found in every AMPA receptor. N1, or the NMDA receptor subunit NR1, is ubiquitous in every NMDA receptor. So you need to have NR1. Again, the new name is GLU-N1. You need to have that in order to form a functional NMDA receptor. So this subunit is always found. Whether or not we have an NMDA receptor NR2A, again, the new name being GLU-N2A, or we have the GLU-N2B, Infempredil will block only the NR2B. We think that this is determining whether the NMDA receptor is found at a synaptic region or it's found outside of the synapse. And so these different subunits here might actually be very important, and knowing that we have it synaptically localized, if we have GLU-N1 and GLU-N2A, that will likely be found at a synapse. If we have GLU-N1 and GLU-N2B, that we can determine also through pharmacology using Infempredil, this tells us that it is likely going to be found extrasynaptically or outside of the synapse. So again, you don't need to worry about having to draw this structure out. However, there are some key features here that I'd like you to make sure that you know. Regardless of any of the glutamate receptors that we're looking at, any of the subunits, kainate, AMPA, or NMDA, all of them have the same topology, which is what is shown here. You don't need to worry about all of the drawing part of it. However, the things that I'd like you to make sure that you know, the N-terminal region is extracellular. This is where glutamate would normally bind in this N-terminal region. And we do have 4 transmembrane domains, but the second transmembrane domain is a little bit unusual. It is called a reentrant loop. It doesn't go all the way through. It is not fully a transmembrane domain region, and this makes the AMPA, NMDA, and kainate receptor subunits unique. I've already told you a little bit about the QR editing site. This is only found on GLU-R2, or the AMPA receptor subunit GLU-R2. We don't see it anywhere else, and this is, again, unique. And the last thing that I've already mentioned is that there is an intracellular carboxy tail, which is also shown on this diagram. And the intracellular carboxy tail contains those phosphorylation sites that might be important for calcineurin, CAM kinase 2, PP phosphorylate as required. So why is all of this important, and why have we sort of taken the long route to getting to what we want to talk about, which is AMPA receptor trafficking and how we know AMPA receptor trafficking actually occurs? So you have to go all the way back 20 years ago, and you have to think a little bit about what was going on in terms of neuroscience. And 20 years ago in neuroscience, this is where molecular techniques first started to really become popular within the field that was just beginning. And one of the first things that people did was to use GFP, this green fluorescent protein, and they wanted to know if after activity they could see any structural changes based on GFP transfection in these different neurons. And it turned out that they could, and this became one of the first reasons why there was interest in using molecular techniques in neuroscience to try to identify some of these big questions that were bothering neuroscientists. How does LTP actually occur? What is the molecular mechanism for LTP? And it was one of those really interesting points in time that I happened to be able to be a part of. And so this was sort of the research that I did with UTN back 20 years ago, surprisingly, when we were looking at trying to figure out if these AMPA receptors could actually move. Nowadays, you learn in first-year biology about these processes known as exocytosis and endocytosis. Again, maybe surprising or maybe not surprising. When I left my undergrad and I was starting off a PhD with UTN, I had no idea what endocytosis was. In fact, no one knew what endocytosis really meant in terms of receptors. And so he had this really easy idea at the time. Receptors could actually be inserted and removed from the plasma membrane. And he had this idea, and he was one of the first individuals in the world to actually have this idea that receptors could move, and we could add more receptors or remove these receptors. And he wanted to be able to prove this. But there was one other catch. So he had a very small team. He was a really young PI, and he had these great ideas. In fact, he still has these wonderful ideas about stroke and recovery after stroke, and he's working on that over at UBC. But he had a really small team, and he knew that there were two other teams at MIT as well as at Harvard that were working on exactly the same idea, that people were thinking about the movement of the receptors for both LTP as well as LTD. And I had never heard of endocytosis. I had joined his lab, and one of the first questions that he asked me was, do you know anything about a cell biological process known as endocytosis? And by the way, Wikipedia didn't exist back then, nor could you do things on your smartphone because they didn't exist either. So I said, just wait 1 hour. I'll run to the library. I'll figure out what I need to look up, and then I'll come back and we'll talk. So again, I had to run all the way from Sick Kids to Gerstein. I had to look up endocytosis. I had to look through these journals. I photocopied a few of them. I ran back and I said, I know what endocytosis is. We can have a chat now. And his idea was, let's figure out a way to prove that receptors can actually move in neurons. Again, it seems like a really easy idea, but in the late 1990s and the early 2000s, this was a really tough thing to do, especially with the challenge of going up against much bigger labs at MIT as well as at Harvard. And so we decided to team up with the group at Harvard instead of trying to compete with them. And we were trying to figure out if we could see endocytosis of the AMPA receptor, because it was easier to prove the removal of receptors from the cell surface than to show that they were inserted. So we actually proved LTD first before LTP, and that's the history of how we know about receptor movement. So a couple of things that I need you to know a little bit about. Triton X100, for those of you who, it might have been a while since you've taken biochemistry, this is sort of a non-ionic detergent that we can use to punch holes in the membranes of cells. It is extracting the lipid to allow the lipid to be removed, and Triton X100 is that type of detergent. And this is sort of what we were doing. We were trying to figure out, how can we see the movement of these receptors? How can we actually watch them moving? How can we quantitate whether or not they move? And how can we prove that these receptors are actually moving? This goes back to that first figure that I showed you after break, which looked at the topology of the AMPA receptor subunits. In the 1990s and early 2000s, people were trying to use molecular techniques to introduce these small sequences that we had antibodies to allow us to visualize the movement of these different receptors. So where you tag the receptor is actually critically important, because sometimes the antibody can bind to it and sometimes it can't. GFP tagging wasn't really an option because when you tag with GFP, it's everywhere. You can't actually see it if it's on the membrane, if it's intracellular, if it's moving up or moving in. We tried that first, and it turns out that if you tag a receptor subunit with GFP, it simply is everywhere. In fact, it looks exactly like this. So GFP is everywhere. I can't see if it's inside the cell. etc. If we were to do this 7 years later, Jennifer Lippincott-Schwartz actually came up with a photoactivatable version of GFP, which you don't need to know the details about, that would have allowed us to do these experiments much more easily, but this photoactivatable form of GFP didn't exist at the time. So GFP tagging was out of the question. These small epitope tags were what UTN wanted to try to do. So we went back to exactly what you have been studying, which is the topology of the different AMPA receptor subunits. And we knew, just like you know now, that every single AMPA receptor subunit within neurons contains what subunit? What's ubiquitous and unique to all AMPA receptors that you look at? So GluR2. So we decided to take GluR2, and this was the sequence for GluR2, and we decided that based on the topology, that it had an N-terminal region that stuck outside of the cell, that we would insert a small epitope tag. This normally doesn't occur within the GluR2 subunit, but we decided to use this hemagglutinin or HA tag, and again, it's very, very small, which means that the nucleic acid would just be 3 times the size of this, which isn't a very big tag. In addition, it doesn't affect the expression, it doesn't affect glutamate binding, it has no functional consequence other than we would have an antibody to HA, and this is normally not found in any proteins that are found within the nervous system, so this would be very, very specific. Doesn't normally exist, we're introducing it, it becomes an epitope. We could use these other ones like c-MYC, also doesn't exist in the nervous system. It's not normally found in neurons, so we could see a ubiquitous tag that normally is not found in the nervous system that we would have an antibody to. In fact, these are some of the other ones. You don't need to memorize any of the sequences or anything else other than to know that it's a small sequence that we can insert using molecular techniques. And usually we pick where we will insert them. So we decided to insert them on the N-terminal region of the receptor subunit. And we decided to pick GluR2 for reasons that I'm hoping that you understand now from the earlier discussion. So before we went any further, because he wasn't sure that this was going to work, we wanted to see first if indeed this epitope tag, we could actually visualize 2 different compartments for these different subunits. So we had tagged both the GluR1 subunit as well as the GluR2 subunit with HA. We had an antibody to the HA tag, and we decided to stain this cell here, which is an HEK cell. It's not a neuronal cell, but it has all of the same machinery. And we decided to stain for the antibody on the cell surface. This told us, does the receptor actually get to the cell surface, as well as permeabilizing the cell using Triton. So we punch holes in the cell with this detergent, and then staining with a different color antibody to see if there's also an intracellular proportion of those receptors as well. And it turns out that when you do that for both GluR1 and GluR2, that we have both an internal population, things that could traffic up to the membrane, as well as a stable population on the cell surface. Do you have any questions about that before we go on? So all of that is just a proof of principle that yes, we can see a population on the cell surface and a population that exists intracellularly. That's important because it tells us that these receptors that are intracellular could, in fact, potentially traffic up to the cell surface if we wanted to examine that later. But this was not the experiment that UTN wanted to try to prove first. So he wanted to see endocytosis. Could you actually see the movement of receptors from the cell surface into the intracellular region of the cell? And so under normal conditions, without stimulating the cell at all, expressing this epitope tagged subunit, just the GluR2 with HA and having an antibody against the HA tag, we were able to show that the population here in green on the cell surface could actually be internalized. And we followed and tracked the movement of these antibody-detected subunits into the cell. So it did show that these different AMPA receptor subunits actually could, in fact, be internalized under the appropriate conditions. So again, just showing you what happens here at the bottom. In yellow would be the tag, the epitope tag. It's sitting outside of the cell, which means that if we have an antibody to this epitope tag, in this case HA, we would then have the antibody tagged to it. The antibody doesn't fluoresce, it doesn't do anything else. It just binds to the HA and only to the HA region. And then what we do is we will allow some of those to internalize and then use antibodies to detect ones on the cell surface, then use tritons, as you're seeing here on the right, to go in and detect the ones that have been internalized, which is why you see the red dots. So we have them all on the surface at the beginning, and then we stain all the ones on the surface, and then we use triton to punch holes and allow a different colored fluorescent antibody to bind to the ones that are intracellular, which is what you are seeing here. Again, I know we just went through a lot of different experimental techniques. Do you have any questions on any of this before we go on? Yes. Why doesn't triton destroy the entire cell? Okay, that's a great question. So on these types of experiments that we're doing here, we actually do what is known as fixation of the cells. So we use paraformaldehyde and we cross-link the amino acid groups together, and that allows for stabilization. If we use triton on a live cell, it would indeed destroy that cell. So that's a great question, but you don't need to know those details. Any other questions? No? So again, this tells us about how to look at receptor complexes that could be internalized using this particular technique. And this is where everyone was stuck. So it's really easy to visualize something that moves from the outside to the inside because we can tag those antibodies get endocytosed, and then we can go back in and detect those antibodies. It's really hard to detect the movement from inside to the outside. And again, it's one of those great things because I got to actually see how UTN's mind worked. And we were at a meeting at the Society for Neuroscience. We had just published our paper. People were asking him all sorts of questions. I remember walking with him at a conference, and we were walking down this one particular aisle that had all these other interesting posters. And then he looked at one poster in passing, and as he was passing, he said, "Bill, I want you to go back and I want you to get all the information on that poster. I don't want anyone else to see that I'm looking at that poster." So I said, "Sure." So I asked him, "Which number were you talking about?" And he said, "That one there." And so I went back, and then I had picked the wrong poster, so I went and talked to UTN, and I said, "Why were you interested in crayfish?" And he said, No, wrong poster, go back." So one of the things that he was really brilliant about was he was able to make these different connections. And he said, "Bill, I want you to figure out how to take that HA tag and then to remove it. Because if we have that, and then we can remove that tag, it would actually allow us to see if a receptor is being expressed at the cell surface." And one of the things that he had saw just in passing, which was amazing to me, so he just literally passed by the poster. He must have had great peripheral vision. He saw that poster, and he immediately made this connection that, hey, we can add thrombin as a cleavage site, the thrombin sequence, and then that would allow for the removal of that HA tag. Now let me take you through why that's important in this experiment. So we cannot easily detect the insertion of new receptors on the cell surface. And again, you need to think a little bit about that or just take it on blind faith that it's hard to do that. And people were kind of stuck. How do we see insertion, LTP, of these different subunits? we had these HA tagged subunits already. We were trying to figure out how to see insertion on the cell membrane, and we couldn't figure out a way to do it. So he decided to actually insert the thrombin cleavage sequence, not thrombin itself, but the sequence that thrombin as a protease uses to attack and degrade proteins. He decided to insert that sequence immediately after the HA sequence that I've shown you already. And what this means is that under the appropriate conditions, we would be able to remove the HA tag in the presence of the protease thrombin. Have I lost any of you yet? So all we're doing now is adding an extra sequence, the thrombin cleavage sequence, to the subunit. Now, this is why it's important to have added that sequence. So one of the things that you can see down here at the bottom is this is an HEK cell, and we did the exact same thing in HEK cells, and then shown to you on the right is the data for neurons. And one of the things that we decided to do was, do we see expression of thrombin, which is in purple, immediately behind the HA tag, which is shown in yellow, just as we saw before. And one of the things that we could do is, if we had this thrombin cleavage sequence and we had these receptors that were already expressed on the cell surface, so this is in a live cell, and in a live cell we know that we have a population of receptors on the cell surface, which is what you are seeing here at the top. And then we also know, based on what I told you earlier, that there is an intracellular population inside the cell, which is shown down here on the bottom. And apologies, these should also have these little purple lines associated with it as well. And now we go in and we add thrombin. Thrombin is not membrane permeable. It will only cleave things on the outside of the cell. This is a living cell, by the way, so the cell is still alive. It still is capable of having proteins move around, etc. So this thrombin is added. This thrombin then goes in and cleaves off the entire sequence. And that means that not only is the thrombin sequence cleaved after the addition of the protease thrombin, but so is the HA tag immediately after it. So HA is gone as well as thrombin. So all of that is removed in the presence of thrombin. Thrombin itself is not permeable. It cannot enter a living cell. And so only the thrombin cleavage sites on the cell surface were eliminated. And then we waited. So is that part clear? So I've eliminated everything on the cell surface. The proteins are still there. They're still intact. There's no HA tag on them anymore because we've eliminated it using that thrombin protease. The ones that are inside, though, because thrombin can't enter the cell, these are protected and these are still intact. Thrombin is there. HA is still found on the intracellular pool. And then we allow for trafficking to occur. And one of the things that we saw is that this intracellular pool, and I thought I had the animation for it, apologies, these will then traffic up to the cell surface. These still have the HA sequences associated with it, and so we could see the insertion of new HA tags because only the ones that were already pre-existing were destroyed, and we were able to demonstrate that we could see trafficking and insertion of receptors that resembled LTP, and we saw that both in HEK cells as well as in the neurons that we expressed this system in. So all of these things together suggested that there is this mechanism for the trafficking of these receptors that allow for AMPA receptors to move into the membrane. That last experiment and the previous experiment, that if they're there on the surface, that they could be internalized, which resembles LTD. So the literature actually began first with LTD and the endocytosis, and later, after about 5 years, we were able to figure out how to examine the insertion that all of you are familiar with now in terms of increased numbers of AMPA receptors. Do you have any questions about this before we go on at all, or anything that isn't clear, or anything I can clear up for you now while it's still fresh in your minds? No? maybe I can ask you this then. How many of you think, not on the quiz, but on the final exam, that you need to understand how this experiment works? That's a pretty good bet. So you have about a month to try to make sure that you understand the details for following receptor movement using the methods that I've just shown you. So I'm going to try to take you through this other paper, because this is actually an important paper as well. We just showed you all of this in a cell culture model. And cell culture models are incredibly important. They're incredibly valuable to us. They give us a lot of information. But a lot of scientists actually have doubts about whether cell culture represents what happens in the real world. And so even though we had this basic mechanism, scientists wanted to be able to demonstrate that does this movement, this insertion of AMPA, actually parallel what we would expect to see as an animal was learning. And in fact, if you look now, this is a few years back, but since 2009, there have only been 10 papers that have been able to do these types of experiments that show receptor movement during a learning process. And I'm going to take you through one of those papers, and I need you to understand some of the different techniques that we've been talking about along the way. So this is a really big person in the field of neurobiology. So if you have the chance to go down to Johns Hopkins and you get to work with Richard Hugener, or you get to work with David Linden, these are two of the biggest names in neuroscience. And some of the key words that are on their paper that came out about 4 years ago are really important ones. So one, it involves NMDA receptors. So again, take a little bit of a pause right now. Does LTP involve NMDA receptors? All of you should be saying it does. By the way, does LTD involve NMDA receptors? It also does. So this is NMDA receptor dependent. This is the movement of AMPA receptors. And this is also demonstrating synaptic plasticity. There's an increase or a change in AMPA receptors, and this is also occurring in vivo. It is not a cell culture model. This is happening in an intact animal, and there are, again, very few labs. Even in Toronto, there might be like one, maybe two labs that could technically try this in Toronto, which is one reason why, if you're doing research, go down to the United States or to Europe, join a big lab that have the funds as well as the techniques to be able to do this. So how do you visualize this in vivo? There's so many neurons out there. How do you actually demonstrate this is occurring in vivo? So this AMPA receptor movement, this trafficking that we demonstrated using epitope tags, how do we actually see this in an intact brain, and how can you visualize the movement? So how do we know that this process is responsible for LTP and LTD, not in a dish, not in cell culture, but actually within an intact brain? How can you actually see an increase in NMDA receptor function or numbers in an intact brain that would therefore parallel what we should see in terms of LTP? So this, again, is a really technically challenging type of experiment to be able to visualize the movement of these receptors. And there are a couple of things that you need to be aware of in this particular paper as well. So they're using a different model system. They're not using the hippocampus. They're still looking at glutamatergic synapses. LTP or LTD, no matter where you're looking within the brain. And one of the interesting things about the mouse sensory cortex is that there is a region of the sensory cortex that has been exquisitely mapped out in high levels of detail that correspond to each of the face. So each of these different whiskers, if you stimulate them, so whisker C4, for example, will stimulate only or activate only certain groups of neurons here in the sensory cortex. E3 will stimulate, if you stimulate the E3 whisker, it will only activate certain other groups of neurons in the sensory cortex, D2, B1, etc. So we have these different types of whiskers and we have different groups of activated neurons that we can go in within the mouse cortex, which we call the barrel cortex because they are organized in these columns that look a lot like these barrels. So this is the model system that Rick Hugener's group is actually using. Again, you can actually go in and you can visualize the activity of these different groups of neurons. So you stimulate the E1 whisker and you can actually see activation of an E1 neuron when you stimulate or you flick the whisker E1, you will see activation of this neuron here in the sensory cortex or the mouse barrel cortex. This is all, again, happening in an intact mouse brain. So that's just the model. How do we actually demonstrate movement of these receptors with different types of stimulation? So if we go back to the cortex, there is a certain group of neurons within the mouse barrel cortex found on layers 2 and layer 3. So the cortex has 6 layers. We're specifically targeting the neurons that are found in layer 2 as well as in layer 3. And one of the things that you can do, and again, it's a really tough experiment to be able to do technically. So you can have a pregnant mouse, and in a mouse, the different mouse embryos are usually all together within the placenta, and you can bring all of these animals out, and you can actually inject in through DNA electroporation. You can inject in a particular genetically modified subunit of the AMPA receptor, and it will specifically target these different layers within. In this particular case, they used a GluA1 subunit, and this GluA1 subunit was tagged with what is called fluorine, and they used a specific form of fluorine, which is GFP. This is sensitive to different forms of pH, and I'll get to that in a moment. And they also tagged the GluA2 subunit with mix, so very similar to what we were looking at before. And they also introduced into this brain DS-RED2. So they have GluA1 with the superecliptic fluorine. They are also injecting in GluA2 with MYC tags, and they are introducing a third gene which is known as DS-RED2, which marks where the neurons are actually located. They're taking all 3 forms of DNA and injecting it into the brain. Electroporation, where you put 2 different electrodes on opposite sides of the brain. As it migrates across this really small brain, when you inject the DNA on one side, it will migrate toward the other, and along the way, as it passes through the cortex, it will actually enter into the neurons found in layer 2 and 3 of the sensory cortex. And this electroporation process is very quick, it's harmless, and it's not at all damaging to the developing embryo. So I also wanted to mention just really quickly, why are they using this superecliptic fluorine molecule, or SEP, superecliptic fluorine molecule? And again, you have to keep this in mind, and I'm going to take you through this in the next few slides as well. So do you remember that we looked at the topology of the glutamate receptor subunits? There's an N-terminal region, and then there's a carboxy terminal region. Normally, when it's inside the cell, the N-terminal region is actually found inside the vesicle. So as it gets up to the surface, the N-terminal goes outside of the cell, but in the vesicle, it's actually found the N-terminal is inside the vesicle itself, and the vesicle, as it gets trafficked up, is actually really acidic. The acidic environment, where it's at pH 5.5, quenches the GFP signal. It's not fluorescent. It only becomes fluorescent when it is no longer acidic. So it stays in the vesicle, it traffics up to the. When it fuses with the plasma membrane on the cell surface, the extracellular environment is no longer at pH 5.5. That's when GFP becomes fluorescent and when we can see it. Inside the cell, In other words, this is not a fluorescent GFP. It's only when it gets to the cell surface that the superecliptic fluorine can actually fluoresce, and this is what is mentioned in purple down here at the bottom. So again, there's a lot of information on that one slide. Do you have any questions about SEP, fluorine, or anything else that I can explain for you now while it's still fresh and somewhat confusing to you? So we are, again, introducing 3 different genes. We're introducing GluA1 with this fluorine that I just explained, the AMPA receptor GluA2 with a MIC tag, and then this other gene, DS-RED2. We have 3 different genes that are being put into the cell, or into layer 2 and layer 3. So again, just to go back, how do we visualize these labeled AMPA receptors? So we've just electroporated. We allow the animal to be born, and then after the animal is born, we can actually use 2-photon microscopy. I was quite surprised to learn the other day that there's now 3-photon microscopy. And the 2-photon tells you about the level of sensitivity. So this can actually allow you to see about, I think, almost half a centimeter into the mouse cortex. And so this is long-distance photomicrographs that are being taken, and you can see these fluorescent molecules within the somatosensory cortex, and they would be sitting on this polystyrene ball. You would have a little window on their skull so that you could peer into it and have your microscope sitting on it, and you can illuminate the fluorescent molecule if it's on the cell surface. And then you can also stimulate the particular whisker that you want. So you can stimulate the E1, the D2, etc., depending on where in the cortex you are looking. So again, this is sort of the setup. This is what they're going to be looking at in terms of whether or not AMPA receptors can actually move in an experience and activity-dependent manner. So this is some of their data, and I won't go through all of it, but I'll take you through different parts of it. So just again, as a reminder, we have introduced all 3 genes, DS-RED2, GluA1 with superecliptic fluorine, as well as GluA2 with MIC. We have put this into the mouse primary somatosensory cortex. There is a map of each of the whiskers on specific neurons within layer 2 and 3. And this represents, again, a barrel. And if we were to deflect either whisker D1 or we were to deflect whisker B1, this actually causes a change in activity that we can measure in terms of blood flow to that area of the somatosensory cortex. So we can measure blood flow. More blood flow means more brain activity, and we can see it over specific groups of neurons, whether D1 or B1. And one of the things that you would see that with prolonged deflection, so as we deflect it more and more, so more activity, just like with LTP, we would actually see more of green fluorescing on the cell surface. Means more of it is being inserted on the cell surface, and we are seeing DS-RED, the outline of the neuron here with the DS-RED. So we're seeing more insertion as we stimulate the whisker. So you stimulate the whisker, you drive more AMPA receptors onto the cell surface, which is why we see more of this occurring. Do you have any questions on this before we go on? So this again demonstrates if you stimulate, you get specific groups of neurons activating, and this causes an increase in AMPA receptors on the cell surface using this SEP fluorine. Any questions before we go on? No? Okay. So again, if you were to continue to do this every single day, and you were to go back and examine the changes in the same area of the brain over day 1, day 2, day 7, 14, 21, and 28, there's actually some really interesting and somewhat controversial data here. So over this 28 days, we get this one spot, because we're going back and examining the same group of neurons every single day over 28 days. We get this same group of neurons, the same group of GluA1 being expressed at the same spot. Is that surprising to you or not? It's either yes or no. How many of you say yes? It's really surprising, blows my mind. How could they possibly publish that crappy data? No, you all like this data then. Do proteins turn over? Yes, no? Should I be calling up one of your biochemistry profs and saying, hey, what's going on with that protein lecture? So proteins turn over. Receptors are proteins. So there's no way for that protein to have lasted 28 days. There's no way that it's the same protein at day 1 that it is at day 28. Normally within the turn over after 2 or 3 days. And yet, we are seeing continually at the same area of the same neuron this expression of GLUA1 on the same part of the brain. So this suggests that neurons preferentially will have expression of AMPA receptors at specific locations all the time. Again, that was kind of new. It was kind of earth-shattering at the time. And this suggests that maybe there is indeed a special pool of receptors that get targeted to a specific region. The other thing to make note of as well, if you stimulate over a few hours, you can actually see structural changes occurring within the spine and the insertion of more of these receptors as well on specific areas. So this again is being able to see in real time as it's occurring the movement and then the long-term stability of these different groups of GLUA1 neurons on the cell surface. So if you track this here at the top and you were to look at it at 1 hour, it's getting a little bit bigger, and at 2 hours it's even bigger, and then 3 hours after you have done that stimulation, it stays really big. You have seen the stabilization and the long-term expression of GLUA1 on the cell surface after repeated stimulation of that whisker. So everything that we would want in an LTP model, you've just been able to demonstrate that here in real time and to have been able to visualize it. So again, just to make sure that there are some things that we've talked about earlier that now I can wrap up for you. So let's go back. LTP actually is NMDA receptor dependent. If it's really LTP, and it should therefore involve the NMDA receptor. We have not talked about the NMDA receptor up until now. We've only been looking at AMPA receptor subunit trafficking, specifically the GLUA1 receptor in that particular model system. So LTP is always NMDA receptor dependent. If we inject in CPP, so do you remember what CPP is? What does it block? Yes. So this is an NMDA receptor blocker. So we inject in CPP into the area of the brain during that time where we stimulate the whisker, and then we want to see, do we still get the accumulation of the AMPA receptors using this SEP fluorine? It shows actually that we do not see this long-term stabilization. At 3 hours, it does not stay there in the presence of CPP. We don't get this increase that we would normally have expected that we just saw on the previous slide. So this suggests that their model is also NMDA receptor dependent. So again, just to make sure we're all on the same page, could I have seen the same results, the ones that I'm showing you down here on the bottom, if I used the MK1, would I have seen very similar results? And basically, I'm asking you Jeopardy music. MK1, NMDA receptor blocker? Yes? Okay, so if you had said in your head, MK1, if I did the same experiment, I would also see a decrease in GLUA1 expression on the cell surface because it blocks NMDA, you would be correct. Would I have seen the same result if I had injected in CNQX? No, because that is an AMPA receptor blocker. So again, here's where knowing some of the different types of antagonists is actually important. So I think I'm going to just leave the last few slides to the next lecture. The next lecture will be a little bit shorter, and we're going to switch things up a little bit. We're going to continue and finish LTP using a slightly different model, and then we're going to talk about CRISPR and its role in potentially blocking LTP within groups of neurons. If you are coming in today to do any of the test viewing, please come directly to the HMB office first, and then you'll be directed to where we'll eventually be meeting. And Kate, do you know if it worked today?
I think it worked.
Okay. Aren't you guys going to make